Hey, everybody. Thank you for coming this afternoon to hear Dr. Amie Settlecowski talk about some of the work that she did as a PhD student at LSU, but then she'll also get into some of the nuts and bolts of what she's been doing here as an NSF postdoc on a project where we're using museum specimens to look at historical changes over 100 years in genomic landscapes in Ethiopian birds. This project, or I should say Amie, is especially well suited to be a postdoc on this project. And you'll probably see why as she starts to go through her first part of her seminar, where she's talking about her research in Bachman sparrows, where she used big databases for distributional data, as well as museum specimens to look at changes in the genomic landscape of Bachman sparrows. She got her undergrad at Tulane, I always want to say Loyola, and then her PhD at LSU. She's been here for about 1.5 years, and I'm happy to welcome her today, and I hope you enjoy her talk as much as I'm sure I will. I'm sorry, I forgot. I needed to read a little statement. The Field Museum acknowledges that it resides within the traditional homelands of the Three Fires Confederacy, the Ojibwe, Odawa, and Potawatomi. It recognizes and is grateful for the original people who laid the foundation of the city of Chicago and the diverse indigenous nations that reside here today. Also, after today's seminar, if you have questions, Jonathan will have a microphone that he'll be walking around with so that people who are on Zoom can hear the questions. And then also, if you're on Zoom, feel free to use the chat or Q&A to put your questions in. Okay, thanks.
Okay, thanks, Ben, for introducing me. I'm really grateful to all of you who are attending. As Ben said, I've been here for 1.5 years, but with typical postdoc life and the pandemic, I haven't really had the opportunity to introduce myself to most of you all. So I'm looking at this presentation as an opportunity to introduce myself and my research to some of the bigger field museum community. So with that, I'm going to get started. I don't think I need to convince any of you all about the wealth and diversity of data that's stored in natural history collections. And that's a good thing because it makes my job easier, because a common through line in my research to date has been using data derived from natural history collections to inform conservation related questions. And so like Ben said, I'm going to talk about some of my dissertation research that was focused on Bachman sparrow. And then I will also talk about the first project that I've worked on here at the Field Museum. So the first thing, like I said, I'm going to talk about is it's 2 projects from my dissertation that focused on Bachman sparrow. They rely on 2 very different types of museum data. And then that will go into something that's related to the bigger Ethiopian montane bird biodiversity project that Ben secured the NSF grant for with Georeferencing. And yeah, we'll go from there. So the first thing I'm going to do is provide some background on Bachman sparrows, since this is a big focus of this talk. They are largely restricted to the fire-mediated longleaf pine forests of the southeastern US. So that's Bachman sparrow on the left, and on the right is a shot of one of the sites that I sampled some Bachman sparrow blood from during my dissertation. In this habitat, the sparrows rely on the forb and grass-rich understory. That's where they nest. And basically, they won't continue to nest there if there isn't a 2 to 3 year fire return interval. That's what's required to maintain their habitat. And so a huge problem for Bachman sparrow has been the loss of this longleaf pine forest through a number of human disturbances like logging and fire suppression. Longleaf pine forests have been reduced to a 3% patchwork of their historic distribution. So in this new map on the right, you see the southeastern US, and the purple shading is the historical distribution of longleaf pines themselves, which tracks with the longleaf pine habitat, longleaf pine forest or savanna habitat. And then the red shading indicates where longleaf pine habitat still existed back around 2010. So as a result of this loss and fragmentation of habitat, the Bachman sparrow distribution is also a patchwork of fragmented populations. And as you can imagine, fragmentation can present a conservation problem. Sorry about that. And in general, habitat fragmentation can provide a barrier to gene flow. The extent that it creates a barrier to gene flow depends on the extent of the fragmentation and also the dispersalability of the organism in question and its proclivity to cross unsuitable habitat. And we can assess the effects of fragmentation in part by inferring population structure, as it can indicate recent isolation. And that's important to identify for conservation purposes because recent isolation can indicate a lack of movement of adaptive mutations and just that genetic variation is becoming segmented across the distribution just as a result of genetic drift. And understanding population structure is also important for conservation frameworks as most of it goes about species by species, and we need to understand distinct population segments in order to drive the allocation of management and conservation resources. Okay, so in addition to human-caused barriers to gene flow, Bachman sparrow is also distributed across the Mississippi River. So the green shading indicates the distribution of Bachman sparrow, and the break in the habitat is the Mississippi River floodplain. There's also been some complicated taxonomic history, of course, in Bachman sparrow. At 1.3 subspecies were described and technically are still described today. But the most northwestern described subspecies, illinoensis, was pretty quickly identified as a subtype of Bachmani and was just the result of people at the time not being aware of the distribution of Bachman sparrow. Today, we primarily recognize two subspecies on the basis of morphological differences. Bachmani is brighter and more rufous, and Estevalis is darker and has these dark black streaks through its feathers. So a previous conservation genetic study of Bachman sparrow was done because of the concern about the fragmentation of its habitat as a result of the loss of longleaf pine forest. It was done with a pretty extensive sampling of Bachman sparrows from across its most contiguous distribution using 18 microsatellites and some Sanger sequencing markers. Interestingly, it uncovered a signal of panmixia or consistent gene flow basically across the distribution, which was really surprising given the habitat fragmentation I described before. And so this is where my dissertation comes into the story. There were basically two possible explanations for the signal of panmixia that were posited. And one of them is Bachman sparrow's association with this ephemeral longleaf pine habitat like I said, they require habitat that's been burned within the last 2 to 3 years in order to nest. Historically, the longleaf pine forest did burn on an average of 2 to 3 years. That's been identified from literature, people who study longleaf pine trees. And so the idea is that with this patchwork of burning that had to occur for over centuries and even longer, perhaps Bachman sparrow and other organisms that have evolved in these types of habitats have evolved better dispersal abilities. And so this would explain having increased gene flow and being able to maintain gene flow across their fragmented habitat. This has been shown on a species basis, a similar surprising signal of panmixia in other species, including not just birds. And I think actually a fellow grad student from LSU, Oscar Johnson, part of his PhD, he did a comparative population genomics study that should be coming out soon that looks at ephemeral habitats in South America and shows this kind of pattern of birds dispersing more in having evolved in that habitat. The alternative explanation, which is not totally mutually exclusive, is that Bachman sparrow experienced a range expansion, which we know or we've seen in the literature. So basically this map now on the right shows the density of Bachman sparrow records from eBird. So Bachman sparrow occurrences documented by anyone from amateur birders to ornithologists since 2000. And so the cool colors indicate fewer occurrences documented, and the hot colors indicate more occurrences documented. So it's a pretty good representation of where Bachman sparrow occurs today. And the dotted black line indicates basically what's come out of this paper that I published, the hypothesis for how far Bachman sparrow expanded in the early 1900s. So in the late 1800s and early 1900s, there were records as far north as southwestern Pennsylvania, southern Michigan, and even Ontario. And this region that extends from where Bachman sparrow occurs today up to that dotted line, I'll refer to as the extraliminal region. The idea that was put forth by the first naturalist to describe this range expansion was that when farmlands moved west during that time, the fallow fields became suitable habitat for Bachman sparrow, and so they expanded into that suitable habitat. And then as that suitable habitat wasn't maintained in that early ephemeral condition, it was no longer suitable for Bachman sparrow, and they retracted to where they are today. Okay, so this is the first project that I'm talking about. Do occurrence records that we can now access as a result of our huge databases that we have now. Do they support the idea of this range expansion? So that's what we're going to look at. And I had a lot of collaborators, of course, not listed, are my PhD advisors, Sabrina Taylor and Jeremy Brown. And yeah, I use data curation from a ton of natural history collections and eBird. Okay, so there's an incomplete understanding of the range expansion and retraction of Bachman sparrow. In part, there's assumptions that are made in today's literature when people talk about the range expansion. They assume that the historical distribution of Bachman sparrow is what it is today. And they assume that the expansion occurred northward, followed by a retraction to the southeast. But this is all based on one study from the early 1900s. And so this map shows in the darker green, the distribution of longleaf pine habitat at its max in the last couple of centuries. And then the lighter green shows another species of pine tree that also exists in a somewhat ephemeral ecosystem. And then the yellow shows our interpretation of the oak savanna habitat that's also now very fragmented. And the earlier study I talked about was based on just this one section of the most northeastern limit of the extraliminal distribution that Bachman sparrow came to occupy. The most common paper that's cited when talking about the Bachman sparrow range expansion is coming from data from that time when it was occurring only within this small box. So there's a lot of factors that complicate our understanding of these range expansions. It's thought to have been occurring during extensive habitat loss, and longleaf pine was deforested to that 3% that remains today by basically the early 1920s as a result of logging and fire suppression. They're also known to occur in other forest ecosystems. Like I said, there was a subspecies that was described from this oak savanna habitat, but that's rarely ever mentioned today in literature that's looking at conservation for Bachman sparrow. So Bachman sparrow also before the early 1920s used open forests like mixed shortleaf and longleaf pine assemblages, mixed shortleaf pine and oak forest, and the transitional oak savanna. So I wanted to look at whether there's more data that could show whether Bachman sparrow historically just occurred in a broader distribution than it does today and has become restricted as a result of all of the habitat loss, or if there was some evidence for the range expansion. And to do this, I gathered records mostly from FERTNet and GBIF and eBird. I also contacted some museums that were geographically important to that extraliminal region that weren't at the time available on the databases. And I reviewed a lot of early naturalist literature from the early 1900s. And I did a lot of filtering of these data sets to make sure we were looking at good records. I also georeferenced every record of Bachman sparrow from the historical databases and from my literature review using the point radius method. So I assigned an uncertainty along with the latitude and longitudinal points that I identified. And for the data that came from eBird, they have a really solid curation process in place. So I used those associated coordinates. Of course, distinguishing between a lack of records and absence is a huge problem. So to facilitate the interpretation of the data that I collected, I went through the same process for field sparrow. I chose field sparrow because it's more common, largely overlaps the breeding distribution of Bachman sparrow historically. It's similarly nondescript in terms of sparrows, and uses early successional habitat as well. So they co-occur also in historical records. There's records of them co-occurring. And so then to look at this descriptively, we mapped the records by the density of records per hectare. And so the color of the cells on the map, like earlier, are go from cool representing fewer occurrences to red representing more occurrence data, and specifically the colors determined by the standard deviation from the mean density per hectare of records from that bird, Bachman sparrow or field sparrow, and time period. So where the map is gray, there are no records, and where there are red splotches, that's where the most records occur in that time frame. So the first map shows all of the records of Bachman sparrow from before the expansion. And notably, there's a cluster of occurrences documented in southeastern Illinois and southern Indiana. Also notably, there aren't occurrences in the region that was described in the contemporaneous literature that initially described the range expansion, but field sparrow was identified and recorded in that area at the time. So it suggests that Bachman sparrow actually wasn't there prior to the range expansion. During the expansion period, there are additional records to support the range expansion. And it seems that Bachman sparrow continued to occur in this region until about 1970. Following the range expansion and retraction, there were some lingering occurrences of Bachman sparrow. And coming up to contemporary times, of course, there haven't been any occurrences of Bachman sparrow for over 20 years. So basically, I showed that the timing of the range shift was consistent with what had been previously written at the time, but the distribution was historically wider than is commonly acknowledged today. That also suggests that there's multiple plausible source regions for the expansion, not just the southeast from longleaf pine habitat, but also the Midwest from oak savanna habitat. And remnant populations likely persisted in the extraliminal region during the retraction, but in smaller numbers. And from a conservation perspective, this is important because restoring not only longleaf pine forests is important for Bachman sparrow, but restoration of other endangered habitats like oak savanna is also potentially important to Bachman sparrow. Okay. So the next thing I wanted to look at was whether range expansion or ongoing dispersal explained the signal of panmixia that had been picked up by microsatellites previously in Bachman sparrow. So again, it's possible that this signal is just a result of gene flow as a result of the range expansion that I just talked about extensively. But it's also possible that Bachman sparrow has a high ability to disperse as a result of its association with these habitats that are ephemeral in nature. So I am going to look at genetic population structure prior to the range expansion and also today using a wider genetic data set. And if the ability to disperse explains panmixia, we would expect to see a lack of population structure prior to the range expansion. And this project took a ton of collaboration and help from just all types of people. I ended up requesting hundreds of toe pads and skin clips from almost 20 museums from across North America, and most of them either sent me them or let me come and sample them. So I'm especially appreciative of that. So the first thing to do for this project was to identify regions to sample consistently through time based on the availability of museum specimens. So it wouldn't really make sense to look at population fragments today that didn't have museum samples from prior to the range expansion. And that range expansion goes back to before the late 1800s. So there is a limited set of museum specimens comparatively from that time. So I identified 5 regions that had consistent Bachman Sparrow specimens from before the range expansion, during it, immediately following it, and then yes, this explains my sampling numbers that I had. And so here you see on the map, again, the green represents the Bachman Sparrow distribution. The black outline polygons indicate the regions that I have temporal genomic sampling for, and the yellow dots represent the localities that I have contemporary genetic samples for. And then the black dots up in the extraliminal region represent museum specimens that were collected from that extraliminal region during the range expansion. So like I said, I also identified modern sites to sample based on where I was able to get museum specimens from. And I went out and collected blood samples from a ton more Bachman sparrows throughout the Southeast. And through collaboration with people who are just interested in the status of Bachman sparrows and the areas that they manage, I also got broader contemporary geographic sampling from a few other places that don't line up with our temporal sampling. So I collected RADCAP data. So basically it's like UCEs, but using RAD loci. So to get at structure or genetic variation that's at a shallower level than you might be able to detect from UCEs, which need to be conserved across a broader group of taxa. So I prepared the historical libraries as regular genomic libraries, and the modern libraries as RAD-seq libraries, and enriched them all for these RAD-seq RAD baits that I had designed based on a preliminary study. The preliminary study was just a RAD-seq study that identified about 3,000 SNPs from across the distribution of Bachman sparrow. So I collected those, I attempted to collect those 3,000 SNPs from across a couple hundred museum specimens and all of my modern specimens, and then went about calling SNPs in the larger data set I collected. We collected a ton of data. Unfortunately, still there was a lot of, we weren't able to collect the full 3,000 SNPs from across every region and time frame. Every region and time frame is represented in a temporal data set of about 950 SNPs. And I also kept a bigger data set of just the modern samples that included even more SNPs. So then I focused primarily on looking at the distribution of genetic variation. So looking at population structure and differentiation, and also other metrics of genetic variation. And I found that there was essentially no population structure or genetic differentiation in Bachman sparrow prior to the range expansion. And this holds basically through time up until the modern period. And also other results looking at the assignment of ancestry using other tools confirms this. Although there are clusters that are identified, they don't correspond to time periods or to regions. Interestingly though, the PCA or the results of the modern data set alone shows that there is some genetic differentiation occurring comparatively. And so I looked further into just those samples in the sites where there was apparent genetic differentiation. And I found that some of the sites are differentiated from the larger cluster of samples. So in part in Texas and Louisiana, in part in Central Florida. So this basically corresponds to regions where I was able to sample from a larger national forest or tract of land, and then also some smaller reserves nearby. And so it seems that there is some genetic differentiation that's occurring in Bachman Sparrow. Also found that there was a significant, a slight but significant decrease in observed heterozygosity, and a slight but significant increase in FST and FIS in breeding, sorry. So although these are slight differences, this is important for the purpose of studying biodiversity through time. Bachman Sparrow looks like it's doing pretty well considering its habitat fragmentation. But if we look at these metrics, we can see that there is a significant decrease in genetic diversity through time, and also a significant increase in genetic differentiation occurring, even if it's slight. So again, there's no significant evidence of genetic structure in Bachman Sparrow. And so the signature of panmixia that's been picked up before and that I picked up again is likely the result of the ability to disperse because of how it evolved in this highly ephemeral environment. But we still find that recent landscape change has impacted the connectivity of smaller segments of the population. And so it's important to consider to continue prioritizing habitat protection and habitat corridors as well between bigger tracts of land and those smaller tracts nearby. Okay, so with that, I'm gonna stop talking about Bachman Sparrow and move on to talking about something broad, which is maximizing the ability to collect whole genome sequence data from 100-year-old museum specimens, and specifically bird study schemes. So this came about because of the project working on with Ben here, looking at the maintenance of genomic diversity or erosion of genomic diversity in Ethiopian montane birds since 100 years ago. Before we wanted to go ahead and start collecting that data, we wanted to make sure that we could collect the most and best data that we could. And so we wanted to do this project first. So historical DNA, like I showed, enables us to understand human impacts on biodiversity. There's the example I just showed in Bachman Sparrow, where there's been very slight changes in genetic diversity and the distribution of genetic variation through time, but we're able to detect them because of natural history collections, and we're able to identify that even near threatened species like Bachman Sparrow that still occurs widely is experiencing impacts from human activity. Bigger study that also looked at just a few taxa, gorillas that got some press, showed that there's been genomic erosion in one subspecies of eastern gorillas that's more significant than a couple other subspecies of eastern gorillas. But moving forward, if we want to use historical DNA to make the biggest conservation impacts, I think the way to go is to do comparative population genomic studies that focus on larger suites of organisms. And doing that requires us to maximize the amount of data that we can get with every sampling event and with every project, because there's limited funding. So although there's been huge leaps in sequencing technology, and the cost is continuing to go down, it's still really expensive to collect whole genome sequencing data from non-model organisms, and especially from museum specimens, because of the nature of historical DNA. which is low in quantity, which makes it susceptible to contamination during the sampling process and during lab work. And it also has low DNA quality. So that ranges from specific molecular changes that happen as a result of time and age and environment, like hydrolysis and oxidation. These changes can inhibit steps of typical library preparations. And also these changes ultimately lead over time to fragmentation, which leads to smaller molecules of DNA and limits sequencing efficiency. So what we know about the best things to do for historical DNA really comes about from protocols that have been established for ancient DNA, mainly for the study of ancient human species and populations. And so from those ancient human studies, they have looked at whether library preparation matters, whether differences in the methods make a difference. And they've found that building genomic libraries from single-stranded DNA rather than just double-stranded DNA improves sequencing results. They've also looked at whether doing single reactions prior to PCR, prior to amplification, or multiple reactions influences the amount of data that's returned. And they've found that fewer reactions does increase the amount of data returned. And that's because with every change between tubes and cleanup that comes along with it, you have the possibility to lose DNA in the process. And also the going back to single-stranded DNA, because of degradation through time, a portion of DNA is going to be denatured to single strands, and that DNA won't be converted into genomic libraries. And when it comes to ancient DNA, a more significant portion has denatured to single-stranded DNA. And then finally, something a little bit more nuanced, but simultaneously ligating sequencing adapters can increase the amount of data you're able to recover, because every molecule that gets adapters ligated to it is able to go through the sequencing process, as compared to some double-stranded DNA sequencing methods that inadvertently end up attaching two adapters of the same kind and don't allow for sequencing as a result. Okay, so something that has been looked at more in historical DNA and museum specimens rather than just in ancient DNA studies is whether the source tissue for where you get DNA matters. This has been looked at a lot, how it influences DNA, the amount of DNA you get from an extraction, and how long the DNA fragments are. The most commonly sampled source tissue for birds is topads, so just a piece of tissue scraped from the bottom of the foot of a bird. Another option are incision line skin clips. Sorry, I lost the slide. But another option are incision line skin clips, which have been proposed as an alternative when the feet include taxonomic characters of interest, or if the birds are small and their feet are particularly delicate and hard to sample. And previous study of just looking at how much DNA you get out of extractions and the size fragments have shown that the yield isn't significantly different necessarily, although topads might produce a little bit more DNA, but the fragment size of DNA in topads is larger. But still, it's hard to know how this is going to influence sequencing because of the issue that I talked about earlier with contamination. Not all of the sequence data we get back is going to be endogenous DNA or DNA from the specimen that we sampled and are targeting with our sequencing. A lot of it is going to be exogenous or coming from contamination from ourselves, the researchers, other birds that are stored in the drawer. If you're doing the lab work in a regular modern lab that does PCR, there could be contamination from PCR amplicons. Okay, so for this project, we wanted to look at specifically how the combination of DNA source from these two most common methods for sampling bird study skins for historical DNA, so how those sources, toe pads and skin clips, and how library preparation methods influenced the amount and quality of data we were able to get back. So we sampled 8 specimens, 4 each from 2 species. Abyssinian thrush is one of our focal species for the larger Ethiopian bird biodiversity project. And then to bolster our sample size, we also looked at American robin because it's closely related, similar in size, and common across North America and also in our collection. We sampled a toe pad represented by the triangular shapes and a skin clip represented by the little trapezoidal shapes from each specimen. And then we also introduced this pre-digestion step prior to DNA extraction. We did a gentle digestion at 37 degrees Celsius, just in the same digestion buffer, trying to remove external contaminants or external exogenous DNA, and to kind of take a look at how that was influencing samples. We took a replicate skin and toe pad clip from 1 specimen of each species. Then we moved forward with extracting DNA from all 20 samples, doing a DNA repair treatment, which aims to get at some of those molecular changes that I mentioned earlier that can inhibit library prep. And then we prepared 3 libraries for each sample. And those libraries were a combination of single-stranded DNA and different tube methods. We did a single-stranded DNA with multiple tubes, single-stranded DNA with just 1 tube, and double-stranded DNA with 2 tubes in the library prep method. And the sequencing metrics that we were trying to evaluate were endogenous DNA content. So what portion of the DNA that was usable was on target for the specimens we were interested in, as opposed to being consisting of microbes or other taxa. What was the mean read length? This is gonna vary more than in modern DNA because there's the degradation of DNA to different fragment sizes. With mean read lengths, the larger it is, the better it is. And that plays out in sequencing efficiency, which is the total proportion of raw data that maps to the target reference genome. So removing PCR duplicates and accounting for that different mean read length. And so what we found in terms of endogenous DNA content is that the single-stranded multi-tube method was significantly better in terms of endogenous DNA than either of the other 2 methods. And there was also a lot of variation in skin clips. So although we didn't find a statistical difference between skin clips and toe pads, we think this is attributed to the variance in skin clips as compared to toe pads. We found something similar for read lengths. IDT was also significantly different from both of the other methods. And in this case, we also found that toe pads had longer read length than skin clips for IDT libraries as well. And we still saw that same variation in the skin clips for read length. For sequencing efficiency, we again found that IDT was significantly better than either of the other methods. So the single-stranded multi-tube method was significantly better than any either. And we didn't find a statistical difference again between toe pads and skin clips here. And we attribute that to the variation in skin clips. So overall, we think the combination of toe pads, of the methods we tested, toe pads and the single-stranded multi-tube method produced the best results. We are interpreting our results as topaz outperforming skin clips when you account for the variation, because it's important to consider consistency. We don't want to have to sample two samples from every specimen to identify, when we're doing these types of studies, to identify the best sample to move forward with. And so in this case, we think topaz give the most consistent results. One question is, why does IDT, which includes multiple reactions as opposed to seriously the single tube, single stranded DNA, why does IDT outperform seriously? I think this has to do with the fact that IDT was originally designed for a different type of DNA, for a different purpose, and the DNA is usually 30 base pairs long. It also includes a two-sided cleanup. So again, more nuanced, but altogether, I think seriously may select for smaller fragments across the distribution of DNA fragments that are available to be prepared into libraries. Notably, seriously, skin clip libraries outperform the IDT method for several specimens, and overall outperform skin clips as well. And seriously is also about 90% less expensive than IDT. So it could be an important method to consider for even more degraded samples than 100-year-old topaz. So the next thing I'm going to talk about quickly is just why we have this, or why we think we have this variance in skin clips. Overall, the variance in skin clips was explained by two specimens where the skin clip always outperformed the corresponding topaz. The first obvious potential explanation could be contamination. But we looked at this by plotting the divergence of each sample to the reference genome, and did find some evidence for contamination in one specimen, but did not find evidence for contamination in the skin clips that repeatedly work out to be better than their corresponding topaz. One of them is represented twice in this graph because it was one of the replicate pre-digested samples. Another option is that they were the specimens where skin clips outperformed the topaz were prepared differently at the time when these were collected. Treating them with arsenic was common, and arsenic is a known DNA polymerase inhibitor. Although previous studies have showed that it doesn't inhibit basically to the extent that you would expect to see these types of impacts. Our explanation is that it's just real variation that comes out from all the noisy factors that go into differentiating individual different specimens. And then finally, the influence of predigestion that we tested just in 2 replicate samples was promising, but not completely clear. And in part, it wasn't clear because one of the specimens we took a replicate from was one of those that had really good results for the skin clips. But we did find in the other specimen that there was an increase in endogenous DNA for the skin clip sample that was pre-digested as compared to the one that wasn't. Okay, so altogether, the takeaways from this are that this particular library prep method with topaz are the best option for whole genome sequencing, specifically for 100-year-old bird study skins, although the other single-stranded DNA method is really promising. Predigestion to reduce external contaminants is also promising. And moving forward, if people want to continue looking at these methods, I think the most important things to look at are the age threshold at which single-stranded DNA methods are warranted due to their being more expensive, and whether keratinized tissues from other taxa, like topaz, come from keratinized tissue on bird feet, whether in other taxa, they're also a better source of historical DNA. Okay, and that's it.
Anybody have any questions in the room?
Well, great talk. Thank you for showing up. And I have a question about the Bachman sparrow.
Uh-huh.
Okay. Why does the sparrow need like this burned area to survive?
So without the burns that come through, that understory just continues to grow, and it grows into like a more extensive mid canopy that's not conducive to their nesting on the ground and moving around.
Oh, they're ground nesters. Okay, that makes
Yeah.
sense. That's it. Thank you.
Thank you, great talk. I have actually two questions, two different ones. One is marginal to what you were presenting, but I was curious because you mentioned some similarities between results related to the Bachman Sparrow
Bachman sparrow, yeah.
and some conclusions from South American study coming out.
Yeah.
My question is marginal because I was just curious about how people deal when you're discussing this kind of issue with the fact the most suitable environment is an early successional one, because sometimes people kind of, oh, this environment is not that worth to preserve, like how people comment this kind of thing, because in Brazil, at least, when we are discussing this early successional environments, conservation, people are kind of, okay, we have to discuss the preservation of Atlantic forest, Amazon, those early successional, they are not that worth it. But my second question is related to that result that it's still in the slide. You consistently show that the topaz sample has a higher sequencing efficiency than the skins, but you are kind of at sometimes it's not significantly. I was kind of curious about what test was that. It was kind of a T of student test that that p-value we were seeing, because I don't know the exact size of your sample, but your box plots are kind of sometimes it's not significantly, but the distribution of the values are not even overlapping. So it's clearly significant to what you are discussing. So I was just curious about this.
Okay, so in response to the first question about, I think whether there's pushback to restoring fire-mediated or disturbed ephemeral habitat, I'm lucky in that Bachman sparrow occurs in the longleaf pine forests, which have been like kind of the premier environment to be maintained by fire in the US. Just it's where these management systems have kind of been developed. And obviously there's other parts of the US where habitat would be better maintained and restored if there were similar reoccurring, consistent fire application like there is in the southeastern US, but there's also less development comparatively to other parts of the country. And so that's part of the explanation. And I would assume it's part of the explanation in other parts of the world too. When it comes to the differences in between topaz and skin clips, the two sample types, I just ran ANOVAs that took into account the source of the DNA and also the method types. And I just never got significant results when it came to source except for read length. We just submitted the paper a couple weeks ago. We'll see if, reviewers say, let's do all the paired t-tests, but just based on, standard statistics, I didn't want to go ahead and do all the paired t-tests without a significant ANOVA result, but we agree. It seems pretty clear that topaz outperform skin clips.
Hi, nice talk, enjoyed that.
Thank you.
Following up on the very first question a little bit, I assume that these feed a lot on insects, at least at certain times.
Yeah, they definitely feed on insects, and probably seeds to some extent. I've seen them eating insects, but they're pretty elusive. So there's not like a ton of study on their diet.
So I was wondering if another thing that would be affected by the change in habitat when you get lack of burns in succession would be changes in the food available to them too. As
Yeah,
well as just the physical habitat for nesting.
yeah, that does make sense. And there has been a little bit of study to show that having some regions of like some denser, not disturbed habitat nearby, people have suspected that that might be important for their prey, like for insects to be living there. And
Sort of a refugium for the insects.
yeah, basically. But the thing is, it needs to be really contained within these fire mediated habitats. So yeah.
Thank you.
Does the red cockaded woodpecker occur in these same habitats?
Yeah, so any conservation for Bachman sparrow has been just adjacent, just as a indirect result of conservation for red cockaded woodpecker.
Would you see them when you were doing your studies?
Oh,
down
yeah,
there?
yeah.
Cool.
Saw lots of red cockaded woodpeckers.
Neat.
Yeah. That was actually how we picked our sampling sites within all of the places that we visited. We were like, where are the red cockaded woodpecker conservation continually managed areas? And we would go to those first because we knew they would be good for Bachman sparrow.
Anybody else? And in the chat, I only see a thank you and from Rosemary Barraclaw.
Great.
All right, thanks so much.
Thanks, John.